Hi everyone! For Chapter 14, we will be discussing a wide array of different scientific methods and techniques we have to study genetics. Some of this you have already heard of before. We've discussed it in previous chapters, but this chapter we will discuss the details of some of these techniques. For example, what are the components, how does it work, etc. So, with discovery of either new genes or new phenotypes like a novel disease, for example, scientists need to study more about what it is, what are the details of the mechanisms, and whether it acts differently in different types of cells or even organisms and species. Hence, scientists have developed many molecular genetic and analytical techniques over the years to answer those questions. We are now able to find out detailed information about these genetic processes, specific ones like replication, transcription, translation, RNA processing, and gene regulation that we have discussed in previous chapter. All those detailed mechanisms we discussed and learned about, and the genetic inheritance patterns we talked about for the entire semester, first half of it anyway, was discovered due to being able to study all the genes and proteins involved using some of those techniques we have already done in lab. Now, for example, staining and preparing specimen to view under the microscope, looking at karyotypes, bacterial transformations, UV-induced mutagenesis, gel electrophoresis, and with that, I'm very sad that we didn't get to experience the whole hands-on part of gel electrophoresis and isolating your own DNA from cheek cells. It would have been a very, very exciting and fun lab, in my opinion. But that's what we have to deal with in times of this pandemic time, stay at home, shelter in place kind of regulations. So now, even though we are studying all these techniques in a genetics class, note that every field of biology will use these same techniques in research, whether it's microbiology, developmental biology, or even ecology. We cannot avoid learning about an organism's genes in any of these different disciplines of biology. If we need to study how related is one species to another, for example, ecology or evolution, we have to compare their genes and see how much of their genome do they share. From a clinical standpoint right now, for example, how we are testing patients to have COVID-19 or not, it's again based on genetics techniques that we're using. Can we find that specific virus's genes in the person according to PCR, which was a technique that you learned and read about in the lab activity last week, and we'll talk about it in this chapter. Kind of like what you did, with the lab exercise last week when you were looking for the ALU positive, or either they have that particular insert in their ALU allele, okay, or they don't. So you were looking for that specific ALU positive band. So that's kind of similar to what they're doing with COVID-19, is that they're looking for the presence of the virus's DNA within the patient to confirm whether they have indeed have COVID-19 infection or not. Okay, now industrially and agriculturally, we are also using a lot of these molecular genetic techniques as well. For example, in the United States, 92% of all corn, 96% of all cotton, and 94% of all soybeans grown in 2017 were genetically engineered to have a higher crop yield and become resistant to some viruses and herbicides. All of those is to be able to allow farmers to farm more crops per unit of space, and also we are able to feed more of the general public because we can increase how much good crops we can yield from the farms when we have these genetically modified products. The whole pharmaceutical industry is highly dependent on development of molecular genetic techniques. Back in 1979 was when a company first developed developed using recombinant DNA technology to make human version of insulin in bacterial cells. When they harvested insulin from animals, which was what they did before the discovery of recombinant DNA, some patients actually developed allergic reactions to, in a sense, a foreign protein that came from some other animal, because it is not our version of the gene or protein product, but as the similar product that was harvested from animals. So the idea back then was that if we can make human version of the gene in another organism that doesn't take up a lot of space and doesn't take a long time to grow and make humongous amount of that protein, for example, in bacterial cells, then it solves the problem. And we did. There insulin now being made in these bacterial cells in companies, and all these diabetic patients out there are now relying on their injections of these insulin to sustain their life. The other protein that's being made in the same recombinant DNA technology is clotting factors. So remember that there are people who are hemophiliacs due to an X-linked genetic disorder, and they need clotting factors, which is what's missing in their bodies due to the genetic mutation. And hence these clotting factors, again, human versions of these clotting factors, can be made in a similar way as insulin in bacterial cells, so that they can constantly be injected with these clotting factors, or once a week or something like that, so that if they were to bleed, they would not bleed to death even with a small cut. So there's a whole biotechnology industry out there because of the discovery of molecular genetic techniques, which we will discuss a lot of these different techniques in this chapter. Last but not least, these techniques are highly essential for studying new diseases or discovering new ways of treatment for diseases. And ongoing research and development of novel cancer-fighting medications, for example, or even though we have discovered all these different genetic mutations that causes diseases like sickle cell anemia or Huntington's disease, we have a way to test whether you have that disease or whether your baby tend to have this disease, but currently there's still no cure for them at this point. We will discuss a bit about gene therapy at the end of the chapter, about some diseases they're trying to cure with gene therapy, and what is going on right now, sort of in a sense where we are at with gene therapy in these few diseases. I think the most common one so far they have been trying to cure is SCID. If you remember, SCID was severe immunodeficient children, okay, where they are born without an immune system, okay, known as the bubble boy disease, okay. So we'll discuss a little bit history of how gene therapy has progressed with that disease at the end of the chapter. All right, so unfortunately, well, for me, unfortunate and fortunate, this is probably going to be the longest chapter yet, because there's all these techniques to discuss and explain through the process. So bear with me, it's long, but yet I find it the most exciting chapter of all. It literally gives me the goosebumps every time I think about, wow, this is where we are at now with technology and what we're able to do with genetics. All right. So what are the challenges geneticists have faced back then and sometimes even now that make studying a gene or a protein's function so difficult? There's a couple of reasons. First of all, remember that only a small portion, probably at 1/1,000,000th of the genome of your 3 billion base pairs in the entire genome actually encodes for a functional product, whether it be RNA or protein that serves a purpose in the cell. We don't know what the other 90% of your genome is doing, but we have to dig for those specific genes that are actually doing something. All these products we're talking about, are invisible to the natural human eye. We can't just look and say, oh, hey, there's that gene and here there's that DNA. Without sequencing techniques we have developed now in modern day, there was no way to see a nucleotide or know when and where a gene begins or stops. So yes, we know, it starts with a promoter and all that stuff, but without sequencing techniques, how do we know that there's such thing as a promoter? Or how can we know where it begins? So even when we are able to locate a gene in the genome, the next thing to think about is how can we separate it from the rest of the DNA? We can't haul around all 3 billion base pairs all the time, from one experiment to the next to look for functions. Plasmids, as we've talked about with transformation, they can only hold some tens of kilobases. So we can't be like, let's put this entire person's genome in the plasmid and see what it does in another organism. It doesn't work that way. It's not like art and craft where we can just take a pair of scissors and cut up their DNA and see what happens. Though we will talk about what's a molecular scissors like and what geneticists have discovered and developed for gene editing. We'll talk about that. So if we are able to isolate our gene of interest, okay, then what? How can we study its effects? Okay, we have to have some way of studying what happens if we take away this gene or if we put this new gene into an organism that doesn't have it to begin with. And if we put the gene into another model organism, how can we ensure that actually does what it's supposed to do? How can we ensure its stability in the organism and not be degraded inside the organism? And let's say that we are able to put the gene into another organism to study. Even in modern days like now, the efficiency of gene transfer is usually very low. So out of 1 million bacterial cells, for example, how can we search for that one cell that has taken up the gene successfully? So we'll discuss some of these challenges and questions and how do we go about solving these problems. This type of challenge, we will see in every new gene or pathogen we want to study. And again, it always revolves around 3 questions. 1, how can we look for and isolate the gene? Okay, sort of like cancer. Why some cancers we can just do a blood test and why some cancer we have to do MRI scan or PET scan to look for them? It's because for some cancer we have discovered that they release what's called markers within the circulatory system that we can look for presence of the cancer. But some other cancer we don't know how to look for it yet. There's no markers that's moving around in your blood. So it's always constantly the same question. How can we look for and isolate the gene? 2, how can we put it in another organism or model organism so we can study it? Preferably an organism that doesn't have the gene to begin with. And 3, how can we screen for the presence of the gene in the model organism? The molecular techniques we will discuss in this chapter will provide us with the tools like scissors and glue in art and craft on how to solve these challenges. These are just analogy, scissors and glue. I'll talk about that when we get to the gene editing part, okay? All right. So whenever we hear the word genetic engineering, okay, we are actually referring to a scientific technique that is used all the time in biological research. We call this recombinant DNA technology, which is a set of molecular technique for locating, isolating, altering, and analyzing specific DNA segments within the genome. And the recombinant word part of that means that this is talking about DNA from 2 distinct sources. For example, we're putting our human version of this gene of interest that we don't know nothing about, and we're putting this gene into a bacterial genome. So we call that recombinant DNA because it's combining genes between humans and bacteria. Now, one of the challenges we discussed earlier was how can we separate our gene of interest from the rest of the genome and insert it somewhere else? Well, one of the key discovery that provide us with the answer is restriction enzymes, okay? The discovery of restriction enzyme. Of course, not surprisingly, it was first discovered in bacterial cells again. Even though bacteria are pathogens, there are good bacteria and bad bacteria. And yet, bacteria is one of the main organisms that has provided us with all the concepts we have been studying all semester long. I mean, transcription, translation, DNA replication, all that stuff was elucidated first from E. coli bacteria cells because they're much simpler organisms to study. So we owe a lot of this knowledge to this little creature. All right, so restriction enzymes are proteins that recognizes a specific sequence in the genome. We call these specific sequence restriction sites. And once the enzyme recognizes a sequence, it acts like a pair of scissors, literally, and cuts the DNA on both strands. Now, again, just like how CRISPR-Cas system was discovered, restriction enzymes was actually discovered the same way. The reason why bacteria has these restriction enzymes was, again, that they defended themselves from viral attacks, just like CRISPR-Cas9 system, and they would use restriction enzymes to cut up their DNA if the virus tries to insert their genomic information into the bacteria. Till this day, there are more than 800 restriction enzymes that were discovered from bacteria, and they cut DNA at more than 100 different unique sequences. And hence, that's why these restriction enzymes have weird names like BAMH1 or EcoR1, EcoR3, EcoR2, etc. There's, because they're all named after the bacteria that it was originally discovered in. So you can tell EcoR1 probably came from E. coli. That's why it's called E. coli. And then BAMH1 actually came from another bacteria called Bacillus amyliliquafaciens. Okay, even I have a hard time to pronouncing that. Amyliliquafaciens is that bacteria's name. Okay, and hence this is why I'm not a microbiologist. Anyway, so the sequences that is recognized by these restriction enzymes are usually pretty short. Anywhere from about 4 to 8 base pairs long usually, and most of them tend to be what's called palindromic. I think we have already mentioned this word once before in another chapter, but it means that sequences are read the same way going from 5' to 3' end on both strands. So, for example, you have GGA TCC here for BAMH1 restriction sites from 5' to 3', and here in the other case, here's a typo from the book. This should be 5'. So from 5' to 3' is read the same way, GGATCC. Okay, so either going this way or reading the sequence this way, they are read the same. That's what palindromic means. So this main table here is just a list of few common restriction enzymes that we use in genetics application. But as I said, we have more than 800 of them discovered, and this is not it on this table, just a few, okay? All right. So now that I mentioned about scissors and glue, and we just talked about the scissors part, which is restriction enzymes. Well, what about the glue part? How can we glue DNA pieces back together after we have isolated them or put them in another sequence? If you looked at the red arrows closely in the last table, you will see that some are pointing at different locations on the two strands. They are not directly across each other. The arrow indicates where the restriction enzymes will cut on the DNA, and for some restriction enzymes, they cut at different spots, okay, from each other, different spots across from each other on the two strands, okay. It's just like this is a cut between G and A here, and Echo R1 will do another cut between A and G on the other side. So this is what we call creating a staggered cut, because the resulting piece of DNA after cutting looks staggered like this, okay, where you have some staggering pattern of the two ends once they're cut. So they have these single-stranded overhangs that create what we call sticky ends, or more properly named cohesive ends. What's special about these cohesive ends is that they contain sequences, of course, that are complementary to each other, and hence they can base pair back with each other to glue the sequences back together. Or, for our genetic analysis case, we want to insert a foreign sequence in between the cut sites and glue those little pieces together. How does that work? So, for example, here's a sequence, one of the sequence where you can cut with, let's say, Hindi3, the restriction enzyme. Here's another sequence that you're going to cut with the same restriction enzyme, Hindi3 again. So now you have two different DNA sequences that both have that same Hindi3 cut site in that staggered cut. And they both have these cohesive ends or sticky ends, and because they're all complementary to each other, technically speaking, other than resticking the blue and blue together and the red and red back together, we can mix it up, where the blue and the red can actually bind with each other and stick with each other and glue together in the end, of course, with the help of DNA ligase to seal the necks within the sugar phosphate backbone of the DNA. But that's basically how we can cut the two sources of DNA that we want to glue together with the same restriction enzyme, because they will create the same restriction enzyme cohesive ends. And because they are complementary, these cohesive sites are complementary in sequence to each other, they can bind with each other. Now, in the end, like I said, we will have to always add DNA ligase to the solution or to the reaction to make sure that we seal those necks in between the sugar phosphate backbone of where they were trying to come together. Okay? So there are other restriction enzymes that don't cut at different spots on the two DNA strands, but at the same spots, okay, where they are cutting straight across each other. So when we go back to this table here, here's one example where you have an arrow cutting between the G and C and another arrow at that very same spot, okay? So this is He3 with creating a blunt end. So this is what we call that the enzyme will create a blunt end fragments instead of cohesive ends, because they don't have those single-stranded overhangs like the cohesive ends, but they cut it right in the middle and you have two blunt ends that's fragment that's created. And there are cases when we need blunt-ended pieces, so we are not limited by sequence matching. Maybe a particular sequence of gene, that we want does not have a restriction site that matches the restriction enzyme. For example, here we're relying on this red gene here having a Hindi3 site, restriction enzyme site. But what if it doesn't have it, and there's nowhere else close by the gene of interest that has the particular site, but it has, for example, He3 or something that creates a blunt ended? Well, that's the only restriction enzyme that we can end up using. So in that case, we will use the blunt ended pieces to connect them together. But, and we will talk about how cloning a specific gene into a plasmid works with cohesive ends in a bit. But note that we can also have blunt-ended cloning as well in the case where our target gene of interest doesn't have those cohesive end restriction enzyme sites. Then we have to rely on blunt-ended ones if there are any. Okay, but we'll talk about how we're able to make plasmid with these cohesive and restriction enzymes. Okay, now interestingly, the length of the recognition sequence, or the restriction site, actually gives us an idea of how many DNA fragments we will likely get if we allow certain restriction enzyme to cut genomic DNA harvest from our cells, for example. Note that the sequence that matches the restriction sites of different restriction enzymes are random. They are located randomly throughout the genome. It's not like, oh, we know this for sure is going to be at, let's say the restriction enzyme site for EcoR1 is going to be at this 2,000 kilobase of your genome. No, we don't know that. Okay, we have random EcoR1 sites throughout the genome. So we will end up with, if we were, for example, to harvest DNA from our cheek cells like your lab last week, and add some EcoR1 restriction enzyme to it and run it on the gel to see what kind of DNA fragments we will have, we will actually see a whole bunch of DNA fragments of different sizes as a result, because they're located randomly throughout the genome. Now, the longer the restriction enzyme cut site is, or restriction site is, the less often that site will appear randomly in the genome, and less DNA fragments will result. Okay. Now, this is based on probability. Okay, it is less likely to find a perfect match of 10 letters in 3 billion letters in your genome compared to just finding that 4 letters of perfect match. It's kind of like, the concept of board game Scrabble. That's why longer words in the game are worth more points than shorter words. It's harder to find or come up with longer words that makes up, 10 or 12 letters compared to coming up with words that are just 3 or 4 letters long. So this is, again, how it can be, the same way with these restriction sites. It's harder to find a restriction site that has 10 base pairs perfectly matching versus a restriction site that has maybe just 3 or 4 base pairs long. We can easily locate those somewhere along the 3 billion base pairs and cut them, whenever they are able to see that cut site, they'll cut it. Okay, and then more cut sites equals more fragments. So that's why shorter restriction site will give you more DNA fragments. Longer restriction sites will give you less DNA fragments as a result. And in some cases, we do want longer fragments, whereas in other cases, we may want shorter fragments. So depending on the application and the purpose of cutting up the DNA, we will use either restriction enzymes that are, recognizing longer restriction sites, or, restriction enzymes that recognize as shorter restriction sites. So how do we typically set up a restriction enzyme digestion? It's all pretty much the same way, where you have a small amount of restriction enzyme and then you add it to a concentrated solution of purified DNA. Why we call purified DNA is because usually we don't take cells and then just dump restriction enzyme site to your cell lysate. After your cells are lysed, sort of broken up from their plasma membranes are popped open, there's lipids, there's proteins within the cells that we don't want. A lot of times we use cleanup kits or techniques that separates the protein and the fat content from your nucleic acids, so that we will only have nucleic acids in your little tube of solution. And hence, that's what we mean by purified DNA. So then your restriction enzyme is not busy looking through all those lipids and proteins for your restriction sites, okay? So that DNA and restriction enzyme are mixed, usually in a given buffer solution, because enzymes need ions and coenzymes as well in order for it to work. So that's why there's those salt solution that contains their patent and different types of buffer that has pH, buffering capacity, and also however much sodium chloride it needs, magnesium it needs, whatever ions it needs, okay? And then you will mix the solution and heat it at 37 degrees for 1 hour. Most restriction enzyme works optimally at 37 degrees Celsius. And after an hour, you're supposed to have all these different DNA fragments that's been cut up by that specific restriction enzyme from the genomic DNA, because technically your genomic DNA are in huge pieces of your 46 chromosomes. But now they're going to be broken up into little tiny pieces that you can run on a gel via gel electrophoresis to look for the different sizes of DNA fragments. Okay, so once scientists discovered how restriction enzymes work, improvements have been made to them so we can cut at specific design places we want the enzyme to cut. Remember that the restriction enzyme that we've been talking about, they kind of cut at random sites that they find those restriction sites. What if we wanted to cut just at one particular area in the genome or two particular area in the genome, so that we have the exact DNA fragment we want, but not a random bunch of small pieces of DNA fragments? So these man-made enzyme are called engineered nucleases. Engineered nucleases is where the sequences were designed to cut genomic DNA at a single specific location, whereas restriction enzymes that we're harvesting from bacteria are just random places everywhere within the genome. So with engineered nucleases, we can direct and tell the cell where to look for to cut the DNA. Now, these engineered nucleases have two parts. One that is the cleaving part, okay, which cuts the DNA, and another part is determined by its amino acid sequence. It does the recognition part that tells the enzyme where to cut. So, for example, if you wanted to direct it to a specific sequence, you would change and engineer those amino acid sequence so that it does recognize that particular part in the genome. Hence, all we need to do is tailor these engineered nucleases to cut at different design location and alter that particular amino acid sequence for different recognition purposes. We can essentially make cuts at any specific DNA sequences we want when we're talking about the usage of these engineered nucleases. Two of which these engineered nucleases that have been made are called ZFNs, or zinc finger nucleases, and TALENs, which are transcription activator like effector nuclease. A very long name. Whoever named that probably have too much time on their hands. Just kidding. But it's ZFNs and TALENs. So this is why I included this cartoon image here to show that we now have all these little cutting tools in our little genetic hands for different purposes of genetic engineering. We have restriction enzymes from our bacteria that we can harvest, maybe like the scissors, and now we can actually engineer them differently to maybe a pliers or a chainsaw, okay, things that cut up your genomic DNA differently, okay. All right, so the more recently developed sequence directed type of DNA cutting other than zinc finger or your TALENs is what we have already mentioned before, the CRISPR-Cas9 system. The CRISPR-Cas system, which is what we named it in the previous chapter. Now we're going to focus on a specific CRISPR-Cas system. So the challenging part of using ZFNs or TALENs is that it was tough to design zinc finger libraries. libraries due to complexities that we're not going to discuss here. It's too long, okay? And the second thing is they often lead to cell toxicity or lethality of the target organisms if we're trying to put these ZFNs or talons in an actual living bacterial cell. And if we want it to continue living for analysis of phenotype usually, then we don't want it to die. So that's the problem with ZFNs or talons. So hence they were able to develop, an even more versatile tool that is both easy to design, less off-target effects so it doesn't kill the cell, and it actually can be used for purposes other than just cutting DNA. For example, we can actually put in particular DNA sequences within a cell. And hence, that's why they developed the they discover the new CRISPR-Cas system, and they have sort of genetically modified this system or mimic this system in their own way so that we can use it for genetic analysis. So the specific CRISPR-Cas system that is most widely used by geneticists to serve as molecular editing tool, kind of like Word document tool, Microsoft Word Office, is the CRISPR-Cas9 system. I know we have already mentioned the basic mechanism of this system, but now we're going to dive a little deeper into the details and things involved or required for this system to work properly. Initially, in the bacteria itself, this system requires two different RNA molecules. One is called the CRISPR RNA. The other is called a tracer RNA. And believe it or not, these 5 letters here again stands for a whole long name that I'm not going to mention here. But it's, shortly known as the tracer RNA. To make it simpler as a genetic technique, geneticists have mimicked these two types of RNA and came up with something called single guide RNA or sgRNA. Which are usually 20 nucleotides long. And this particular single guide RNA is what we're relying on to search for our targeted gene of interest. So you can think of the CRISPR-Cas9 system kind of like using the Control F find tool in your either Adobe document app or your Word processor. Okay, so you have a 300-page electronic book. You want to look for a certain specific word or phrase. Okay, so you pull up your Control F, which is your find tool. Then it's going to search your entire book for all those locations that has that specific word you're looking for. So this is what CRISPR-Cas9 is doing. Instead here, we're looking for a gene with that specific 20 nucleotides out of the 3 billion base pairs you have in the genome. And now, of that 20 nucleotide sequence in the single guided RNA, 8 to 12 of them are what's called seed sequence, which are the most important for pairing purposes. Okay, so that's why I said it's kind of like control find, because you're control finding that particular gene within your whole encyclopedia of genes. Okay. So the next thing to do after we have our single guided RNA is combine it with our molecular scissors, which is the actual Cas9 enzyme protein, and form what's called an effector complex. Okay. Now, one requirement in the target DNA sequence for this CRISPR-Cas9 system to work is that it needs to have what's called a PAM sequence. Okay, PAM sequence, again, it stands for something long, protospacer adjacent motif. Okay, so this PAM sequence has to be there, sort of close by your targeted gene of interest in order for that effector complex of CRISPR-Cas9 system to bind. If the target area of DNA does not have this PAM sequence, then you will need to use another CRISPR-Cas9 system from another bacteria that recognizes a different PAM sequence, so that might match your specific target area. Remember that this system was modeled after bacteria's way of attacking foreign DNA. So requiring a PAM sequence was one of the safety measures the organism has evolved to have to ensure that the bacteria doesn't cut up its own DNA as well with the CRISPR-Cas9 system, okay? So once the effector complex binds, though, then Cas9 will unwind the specific region of DNA like helicase does in DNA replication, and allows your single guide RNA to base pair and find the specific sequences, and then Cas9 will cut the specific sequences on both strands, making a double-stranded break. So again, this diagram here puts you in a good perspective of what's happening. Okay, so again, you have your single guided RNA bind with your Cas9 protein to get your effector complex. Effector complex looks for the PAM sequence in your target DNA sequences, and then once your effector complex binds to that PAM sequence, then your very important seed sequence of your single guided RNA will bind to a particular gene that you're looking for and cut that specific portion of the DNA sequence. And another hint here for answering, those short answer questions I have on the exam is if you can understand the basis of these diagrams, it tells you pretty much what's happening and you can answer those questions pretty easily. I'm not looking for completed English quality essay that you turn into an English professor. I'm looking for key points that is essential for that process, and that it shows me you understand the main parts of that particular process or mechanism. Okay. All right, so what happens after cutting the DNA is what makes CRISPR-Cas9 system so unique and a powerful molecular genome editing tool. Not just a cutting tool, but an editing tool where we can insert some sequence that we want inside the genome. So after a double-stranded break is introduced into the target DNA sequence, the normal cellular DNA repair mechanism will try to fix this break right here. There's two ways that the cell can repair the break, but both will still result in a non-functional gene, regardless whichever one the cell decides to use. One is called non-homologous end joining, which is where they join together the two ends of the DNA without any template. It's just, here's a blunt end, here's a blunt end. Oh, let's join them together, smack next to each other, okay? Now, because you're not using a DNA template as a guidance in this type of DNA repair, okay? The short for this type of DNA repair is called NHEJ. And when you're doing this type of repair without template DNA, you can produce small insertions or deletions at the break site, okay? And remember, we just talked about from last chapter that indels will create frame shift mutation that can disrupt the coding sequence, and hence you end up with a loss of function within the protein, hence disabling your gene of interest. So that's how you end up with a non-functional protein, but yet you don't cause crazy things going on in your cells because there's a broken piece of DNA somewhere, okay? The other way that the cell will try to repair the break is called homologous recombination, or HR for short. So homologous recombination is when a donor piece of double-stranded DNA with complementary sequences to the ends of the break that was made by Cas9 can be introduced as a DNA template. That's us, okay, providing the cell with this, hey, here's this good piece of DNA we want you to put into the sequence. It's good for you, okay? So that's the donor piece of DNA that we can put and insert that desired sequence within the genome. Now, of course, we hope for it to do this, but it is not a very efficient technique, and usually not a high amount of cells ends up doing this homologous recombination, and hence that is why we still need to screen for particular cells that actually did this in their genome. All right. So in the end, you will end up cutting the original target gene of interest, okay? and possibly put in another sequence that one wants to put back into the genome. Now, this, whatever you put back, can be the original wild type version of the gene to see if it fixes a certain disease's phenotype, or introduce another mutation to observe its effects. Okay, so that I'm talking about these two. You're either deleting or duplicating a certain part of the gene that makes it non-functional, or you're correcting it with your DNA template back to the wild type version, or some other mutation that you want to do, maybe a second suppressor mutation or something. Okay, so in either cases, the CRISPR-Cas9 system has been under constant redesign and remodeling so it serves as an even more versatile tool for genome editing and possibly used to cure genetic diseases one day. That's the hope anyway. And we'll discuss some of the other hopes of genetic gene therapy. For example, if we know a disease results from one nucleotide deletion, okay, we're talking about hopefully one day this is how we can use CRISPR-Cas9 in diseases, okay? Maybe we can say, hey, don't worry about it. We know exactly where that one nucleotide was deleted. We can search for that specific location of the deletion with the single guided RNA, and then use the Cas9 system to cut and insert the missing nucleotide in, and ta-da! No genetic mutation anymore. Okay, so that is the easier said than done kind of deal in terms of what we hope to do with this system. But regardless, whether we get to the clinical aspect, cure aspect of this Cas9 system, we're currently using CRISPR-Cas9 system all the time in research to figure out how we can study certain diseases, or figure out how we can study certain gene. We want to know what is the phenotype if we take out this gene in a cell. And just because it provides a much quicker way for knocking out a gene within a cell. All right, in any case, we're still, of course, far away from that ideal situation of ta-da, here's that gene mutation and then it's gone, But now we have at least a molecular tool such as these three we have mentioned earlier to discover, study, and possibly fix the genes in the cells. Remember that after cutting with any of these three systems, either with zinc finger, TALEN, or CRISPR-Cas9, we can either just disrupt the gene sequence and make it non-functional, or we can substitute with another sequence, one that can either introduce another mutation or replace the entire segment of the gene, such as a wild type version of the gene, so that we can swap out the mutated gene with the wild type version of the gene. Now, a lot of times after cutting our DNA of interest, going back to the restriction enzyme and all that stuff, we may want to confirm that the cut was made correctly as we have planned and designed, even when we're talking about the use of those engineered nucleases. Since we can't visually see sequences with our own eyes, or even DNA for that matter, we need to rely on a different visualization tool for DNA, which is gel electrophoresis. And we have already talked about that in the past in the lab. So any gel electrophoresis go, whether it's for DNA or protein, it is to separate the molecules based on size and charge. Okay, based on size and charge. We have already introduced this tool in our last two lab exercises, and the gel itself is usually made with a molecule called agarose. Agarose is a polysaccharide that is harvested from seaweed originally. It comes in a powder form, and you can dissolve it by heating it in a specific salt fluid buffer. Now, why we have to dissolve it in this fluid buffer is because this salt buffer is required in order for the electric current to run properly through the gel in that gel apparatus you saw in your lab last 2 weeks ago. So once, it's dissolved, we need to make sure what's in the gel, the salt concentration is equal to the buffer that we use to, sort of fill the gel boxes with in order for the electric current to run through the fluid, in a sense, run through your gel as well. Okay. And once the agarose is dissolved, the solution can be left to cool. That solidifies into a gel-like matter. Okay. Now this is the fun part of the experiment. I said that I'm sad to have missed, because I remember when I made my very first agarose gel after lab, I would play around with it like hardened jello, and it's just fun, kind of like how people like to pop those bubble wraps. It gives me that same feeling when I break the little agarose gel into tiny little pieces. Okay, well, that's just my little spiel of not having hands-on lab. Anyway, so the gel itself acts like a sieve. Okay, we call it molecular sieve, okay, that will sort any molecules that travel through it by size. Bigger things will travel slower through the sieve, while smaller things will travel faster. Now, DNA travels in one direction on the gel because it is negatively charged, okay, so negative charged DNA particles, because they have what the phosphate groups in their DNA backbone, and those phosphate groups are negatively charged. So like a magnet, that negative charged piece of DNA will travel towards the other side of the gel when an electric current is applied to push the molecules along the gel. And that's why you always have a negative electrode on one side of the gel and positive electrode on the other side of the gel, and DNA will migrate downwards toward the positive side of the gel because DNA is negative charge. Now, there have been cases where students, or even graduate students, they would mix these two electrodes up, setting up the gel, and your DNA end up migrating out of the gel into the fluid surrounding it. So there goes your DNA sample. Bye-bye. Okay, so we have to make sure that these electrodes are put in the right direction to make sure that your negatively charged DNA is traveling towards the correct positive electrode. Okay, so the first well, that's, usually these wells we call are a set of holes that we create within the gel to load samples into. So these little flat gels that we're making actually have little holes in them at the top, like this, rectangular holes, and we call those wells. And the first well usually contain what's called the marker sample. Okay, the marker sample, or what we usually refer to as the DNA ladder if we're looking at DNA samples. There's also protein ladder as well for protein samples, but its purpose is to serve as known standard length sizes of DNA that we can correlate how far the bands are migrating to the size of the DNA fragment. So for example, if this is your molecular standard, okay, or marker sample, this is, let's say, 10 kilobase, okay, and this is 7 kilobase pairs, okay. We know that because that's our standard. So whichever DNA that has migrate, maybe right about this one, it's kind of close to the 7 kb. So one might estimate it to be a 6 kb fragment, and this would be close to the 10 kb fragment. So maybe this is a 9 kb fragment. There's actually systematic way and computerized program where we can actually measure the distance from the band to the top of the well, measure it exactly, and then do a mathematical comparison with the distance that's traveled by your standard size letter, and calculate to estimate the size fragment of this DNA fragment in the gel. Okay, but we can't go through that now because we're not running the gel, but that would have been what we would do in lab had we actually run the gels ourselves. Okay, since DNA is colorless, in order to visualize DNA fragments, we have to add fluorescent dye either to the gel itself or DNA samples. Remember we talked about aethylene bromide in the last chapter as a mutagen, okay? It is a DNA intercalating agent, and we said, it actually sandwiches itself between the two DNA strands, okay, so that it creates some sort of insertion and deletion deletion for mutagens case, but in this case, we wanted to sandwich in there because ethylene bromide is a chemical that will fluoresce when you subject it to UV radiation. So that's why it acts as a good candidate for DNA dye, okay? But a lot of labs are now refraining from using it due to its carcinogenic effects, as we have discussed in the last chapter, and hence other colored or fluorescent dyes are being used instead to see DNA bands like methylene blue, or what I used to use in my research lab was called gel red. But both of these cases are chemicals that are not carcinogenic and not toxic, so that we can still see DNA bands without potentially harming ourselves. Regardless, usually the fluorescent dye would glow when bombarded with UV light, just like this, okay, in the case of ethylene bromide or the gel red that I've used. And now we are able to actually see the bands. And in a research lab, a lot of these UV light boxes are actually hooked up to a digital camera intrinsically, and it will allow for a convenient way of capturing the gel image as data for record. Of course, we don't have this here in school setting, but we can take picture with our phones as well, making sure we're protecting our eyes from those UV light. That's why there was that plastic cover on top of the UV box you use to see your glowing bacteria back when we had that lab. Okay. So let's finish with PCR and then we'll have a break, okay? We have discussed how we can cut up and, in a sense, isolate our gene of interest from the rest of the genome. We have discussed how we can visualize and confirm the size of the DNA fragment we want to cut. So the next thing we want to address is having enough samples to begin with for doing anything else afterwards, because, after we cut up the DNA, we probably want to do something with it in order to study it. So the next thing we want to address is how can we make sure we have enough DNA samples. If we don't even have enough of that targeted DNA sequence to begin with, how can we do any analysis with it afterwards? A tiny amount of DNA is usually not enough to see on a gel. Those bands you saw on the gel electrophoresis earlier, well, those were at least from micrograms of DNA. But what if you only had nanograms? You can't see those, let alone if you just had one single piece of DNA molecule. We can never see that on the gel. And hence, we need a way to amplify a specific sequence and be able to make millions of copies of that specific sequence. This has to be done in a method that has high fidelity, meaning that it will not make mistakes. It really clones your original sequence 100% correct, and high efficiency, which means it won't take all day or 24 hours just to amplify the sequence, or else it will take too long for any of these to work, okay? It has to have high fidelity, high efficiency in order for it to be usable in genetic settings. So here comes the development of our DNA amplification method called polymerase chain reaction, or known as PCR. We kind of talked about this in the lab exercise, so I'm going to explain through it this time, okay? So PCR was a method developed by a biochemist named Kary Mullis in 1983, who was studying the hemoglobin gene in sickle cell anemia. He actually won a Nobel Prize for it in 1993 because of how much of an impact and change PCR has revolutionized our genetic techniques. That's why he won a Nobel Prize. And it's actually highly versatile, where we're using PCR for a whole variety of reasons other than just studying a gene, okay? So again, we're now heavily relying on PCR, not just for research purposes, but disease identification in patients. For example, right now with COVID-19, the test they're doing in patients is the PCR reaction that makes sure, to look for that particular band corresponding to the virus DNA. Crime scene investigation is also relying on PCR methods, and even paternity disputes rely on PCR methods. polymerase. Okay, we'll discuss how DNA profiling works with PCR at the end, close to the end of the chapter, and how it works to identify a suspect or relate a suspect to the evidence collected from the crime scene. Okay, so the technique used being for a PCR is based on using DNA polymerase, okay, because it is doing what happens in a cell. Remember, in a cell, DNA polymerase is doing DNA replication. So now we're trying to do DNA replication in a tube versus a cell, and we're mimicking what's happening inside a cell during DNA replication. And we're trying to duplicate our specific gene of interest over and over and over again numerous times, achieving amplification of a single piece of DNA molecule to millions and billions of copies. Think about a tiny piece of skin or hair follicle that was picked up from a crime scene that happened weeks ago. Okay, some DNA may degrade over time. Things happen during that week. So the amount of DNA that is picked up by a forensic investigator can be very, very, very tiny. Maybe even just a few molecules of DNA, which now we can actually amplify it with PCR, okay? With those small little few molecules of DNA we pick up from the crime scene, we probably can never visualize that as a readout. But if we can copy that one single piece of DNA over and over and over again with high fidelity and high efficiency, like a copy machine, then we can amplify the signal and be able to see it as a DNA band on a gel. And that's what the PCR does, okay? So the materials required for PCR are your DNA template, which is that target sequence we want amplified. We never want to amplify the entire genome. We will just want to look for that specific gene we're looking at. A pair of single-stranded primers, just like how in DNA replication, you had to have primates to lay down primers, RNA primers, to start the process. In this case, the same thing. DNA polymerase is going to look for that free 3' hydroxyl group from primers. So that's what we have to add and genetically make or engineer these primers to be added to your mixture of solution. Okay, so these primers are usually complementary with your template sequences, and usually there's a pair of them, meaning that there's a forward primer and a reverse primer. Okay, so what happens is you have your DNA template right here. One recognizes something here and one recognizes something downstream. Okay, so that you have a forward and reverse primer that tells your DNA molecule when to begin replicating and when to end replication, because technically speaking, we have nothing to tell it to terminate the DNA replication like a cell does. So that's what reverse primer will do for us. Of course, we can't forget our DNA polymerase enzyme, which is adding those nucleotides to the template. And we have to add your 4 types of DNTPs, which is your adenine, thymine, cytosine, and guanine. And then, of course, you need your buffer, which contains salts required for the reaction to proceed. And also magnesium ions are also critical for DNA replication to happen correctly, or for the DNA polymerase to work correctly. Okay? So the process of PCR actually was refined later on by replacing the DNA polymerase from E. coli. Okay, originally we started with E. coli polymerase because that's where we discovered DNA replication from, and that's the main bacteria that they were working with back then. But what happens with E. coli DNA polymerase is that we require a step called denaturation within PCR reaction, which means we need to boil your sample. Literally, the PCR sample will be heated up to 90 to 100 degrees Celsius. Okay, so that denatures the DNA. Well, what else denatures? Proteins. So every time the DNA is being denatured in a cycle of PCR reaction, your polymerase is also denatured. So what scientists end up having to do is keep going in there and add fresh polymerase after every cycle of repeats. So instead of doing that, which is very cumbersome and time consuming, it defeats the purpose of having PCR, is they substituted the E. coli polymerase with a special type of polymerase called Taq polymerase, and this is also discovered in another bacteria called Thermus aquaticus. And these bacteria are actually found in hot springs, okay, of Yellowstone Park, okay. So those hot springs, of course, is very, very hot, okay. Some of them are at 90-degree temperatures. So that means for those bacteria to be able to survive and multiply within the hot spring, their DNA and, their proteins and enzymes are properly made in a special way that they can withstand such high temperature. So Taq polymerase is their version of the polymerase for DNA replication. So once they have substituted the DNA polymerase from E. coli with Taq polymerase, now we are able to just add everything from the beginning into a little tube like this, and then put it into a PCR machine, let it run for a couple hours, and we're done with it, okay? Never have to touch it again. So that was a very innovative and big improvement to the PCR reaction. So the process of PCR goes through rounds and rounds of 3 of these following steps, okay? So step 1, like I said, your DNA needs to be denatured into 2 separate strands. So the first step is you program the PCR to heat up to 90 to 100 degrees Celsius to denature and separate your 2 DNA template strands. Now and then, your PCR will be programmed to cool to about 30 to 65 degrees Celsius to allow the annealing of the primers. Note that it sounds very easy here, where here you go, here's the recipe, input it into machine and you're done with. In reality, for every new set of primers that you're using to identify different genes, you actually have to optimize these conditions. That's why it's 30 to 65, because for some primers it's better at 50, for some primers better at 32. Okay, so what researchers usually have to deal with is try different temperatures of annealing and try different concentration of primers as well for this step. So that's why there's this wide range of temperature right here in the second step. And third step, once the primers are annealed correctly to your DNA template, then we allow DNA replication or DNA synthesis to happen using your DNA polymerase, in this case, Taq polymerase. And that is heated to 72 degrees for allowing that particular polymerase to work correctly. Okay, then after that, you're done with the first cycle and your PCR machine goes back to step 1 again, heat up all the way to 90 degree and then cools down to 30 to 65 and then heat back up to 72. So it goes through this same step 1 through step 3 multiple times, almost 30 or more rounds of it. After 30 or sometimes 40 cycles, it depends. That's another thing that researchers have to play around with, is how many cycles of PCR reaction do you have to go through or ask the machine to go through in order for you to get enough DNA sample, okay? But technically speaking, after 30 or more cycles of PCR, you would go from 1 piece of DNA to 1 billion or more pieces of DNA molecules. And this diagram here kind of shows you what happens step by step. So this is that first step right here, going from double-stranded DNA template and denaturing and separating into the 2-strand. And the second step is annealing of that little tiny primer we have. And hence now DNA polymerase is held at that 72 degrees and allow for new DNA synthesis to happen on your DNA template. And once that's done, then you go through second round. Okay, so this is first cycle. And this is your second cycle. Denatures again, primers bind, and then DNA is synthesized. Okay, so, and then it happens a third cycle. Okay, denatured your DNA template, and then your primers are annealed, and then DNA is synthesized, and goes on and on and on and on for however many cycles you set the machine to happen to. Okay, and that's how PCR work. I will include an animation video of this on eCampus for you to follow through. It's easier sometimes just to visualize it pictorially in a video. And I will take a break here and continue the rest of this chapter in part 2 of the lecture video.